Good afternoon and welcome everyone to today's Horizons of Engineering, Medicine and Industry seminar. My name is Jeff Sewardson. I'm a professor of biomedical engineering and I'm the vice chair for BME Clinical Collaborations and Industry Collaborations. And today's Horizons seminar, or HEMInar, is a great example of what we hope to accomplish in this seminar, which is to present science and translation that's at the interface of academic research, clinical impact, and research and development from the industry sector. And so today's speaker is a great example of that. And I'm especially happy to see in the audience people from every dimension of the scientific endeavor that I just mentioned, including not only academic researchers and some folks out there that I see from industry or technology transfer, but also clinicians. And so something that I think is really special about this seminar is that it's one of the times that the researchers and clinicians, and by the way, those who do both, can come together and share perspectives. And of course, that's something that we cherish very much at Johns Hopkins. I want to thank Winston Timp for helping to organize today's seminar. And with that, ask Winston to introduce today's speaker.
Thanks, Jeff. Yeah, it's my pleasure to introduce today's speaker. We're particularly privileged and happy to have Ron Mazumder come to talk to us today. So Ron's an alum of Hopkins. He got his bachelor's here and then followed up with a PhD at University of Maryland and more recently got an MBA at Lehigh. He's worked in a couple of different pharmaceutical companies, leading development of different questions, especially focusing on things that are oriented around biomarkers and being able to predict and identify things for diagnostics. He's worked at places like Janssen with Johnson & Johnson. He's worked at Merck, but most recently, in 2016, he joined Genentech as the VP and Global Head of Oncology Biomarker Development. There, he's leading a team of computational, translational, and biomarker scientists to develop sort of predictive biomarkers and companion diagnostics to learn more and be able to predict better questions in cancer. He's also tried to implement some of these novel platforms into clinical trials to get a better understanding of resistance mechanisms and how these drugs are applied. Today, he's going to tell us some of that. He's going to tell us about how the impact, his seminar is titled The Impact of Computational Biology on Cancer Drug Development, New Insights and New Paradigms. And I'm excited to hear about some of the work that they're doing at Genentech and some of the stuff he's going to tell us about how they're integrating computational biology approaches, technology development, and data science to get a better understanding of disease biology and pharmacology, including questions of like single cell RNA, spatial RNA, circulating tumor DNA, and then the follow-up data analysis of that to illuminate sort of novel targets and pathways for drug development. With that, I'd like to invite Ron to sort of take it away and tell us a little bit about what he's come to share. Ron?
Thanks, Winston, for that great introduction, and thanks for the opportunity to speak. I think first of all, I'll just say it's a very exciting time to be in pharmaceutical drug development these days with a huge convergence between biology, technology, and data science. And I'm going to show you a few vignettes of some of the research activities we have that hopefully will demonstrate that point. So I think we are focusing in Genentech on 4 pillars of drug development in the future. One is really, can we develop a really deep understanding of human biology so we can really understand disease and therapeutic vulnerabilities? Secondly, can we bring to bear single-cell technologies in a high-throughput manner, whether it's single-cell RNA sequencing, TCR sequencing, ATAC sequencing, where we can look at chromatin accessibility, perturb-seq, and site-seq, where we can look at transcriptomics as well as epitomics, as well as can we look at new modalities, so not only small molecules as drugs, as well as antibodies, but can we also look at gene therapies, cell therapies, antibody drug conjugates, and bispecific modalities as well. And lastly, advances in machine learning and AI. And we have our new head of Genentech Research and Early Development, Aviv Regev, who formerly was at the Broad Institute, and she's really pushing for a lot of these types of modalities. And I'm going to focus on my talk today on the first two of human biology and these massively parallel high-resolution methods. So I'm going to go into a little bit about how we're looking at resistance mechanisms in cancer immunotherapy. I'm going to talk about TIGIT, which is a new target for cancer immunotherapy. I'm going to talk about how we're trying to understand tertiary lymphoid structures and their role in dictating response to cancer immunotherapy. Some transcriptional programs that are initiated by tumors, depending on their mutation subtypes, and a little bit about circulating tumor DNA in the adjuvant setting to help direct some of our therapies to patients who would respond. So I'm going to start with the hallmarks of cancer, and Doug Hanahan and Robert Weinberg published a very nice article about 20 years ago now about the 6 major hallmarks of cancer, covering apoptosis, angiogenesis, growth factor signals, replication potential, and tumor invasion. And they published in 2011 an update to that they call the next generation of hallmarks. And they one of the emerging hallmarks was avoiding immune destruction, and that's going to be the focus of my talk today. In the interest of time, I'm not going to have time to talk about one of the enabling characteristics of tumor-promoting inflammation, but suffice it to say that we're seeing some fascinating data in our studies as well as in the scientific literature about how some of these oncogenic drivers actually create a very immunosuppressive microenvironment that is much more pro-tumorigenic. But today I'm going to focus really on the avoiding immune destruction. And to do that, I'm going to use this framework that was first put together. It's called the cancer immunogram, first put together, proposed by Ton Schumacher and Anthony Rebus. And this is a modification of that was proposed by John Charles Soria just a few months ago. And he talks about the many factors, as you can see here, that are involved in dictating response to cancer immunotherapy drugs. And in the interest of time, I'm going to focus on just 4 of them that are shown here in the box. So having a favorable tumor microenvironment, soluble inhibitors, as well as new targets. So I'm going to start with a framework that was developed by my colleagues at Genentech, Dan Chen and Ira Melman, called the Cancer Immunity Cycle. And I want to focus your attention really on steps 5 and 6 and 7. Those are the ones I'll be mainly talking about. But step 5 is really infiltration of T cells into the tumors. So that is obviously a key step, and I'm going to focus on some of my initial talk on that step. And what I mean by that is we and others have published that there are 3 basic immunophenotypes that are present in tumors. If you look at the pathology images on the bottom here, you have infiltrated tumors where the T cells can actually infiltrate into the center of the tumor. You have the excluded immunophenotype where the T cells are largely on the outside, the perimeter of the tumor. And then you have what's known as the desert or cold tumors where there are no T cells present. And the reason why this is important is because, as you can see here, the prevalence estimates by our data and others is that you can see, depending on the various tumor types, renal cell carcinoma, non-small cell lung carcinoma, urothelial cancer, colorectal cancer, prostate, and glioblastoma, you can see very different percentages. And you can see that most of the success we've had in cancer immunotherapy are not surprisingly in cancers that have high percentage of inflamed, whereas glioblastoma, where we've had much less success, prostate and colorectal, are by and large more excluded and immune deserts. So the question we ask ourselves is, first of all, how does this relate to survival and to outcomes in cancer immunotherapy? And it's very important because, as you can see, this is called a Kaplan-Meier curve. For those of you who are not familiar with looking at Kaplan-Meier curves, I'll just orient you. On the x-axis is time, so that's overall survival. On the y-axis is the probability of survival. You can see here the 3 different graphs. If you actually had a perfect drug, you'd have patients who basically they would stay at the 1.0 line. You would have no deaths here. But you can see what happens over time is that you do have patient deaths, unfortunately, and the curves come down over time. Typically, you'll see what's called landmark survival at 2 years or 3 years, and you can see with the blue curve, for example, you have about 20% survival after 24 months. With some of the other curves, it's a bit higher. But what you can see here is that the curve that is the inflamed curve, those patients actually have better survival in terms of not only percent surviving at 2 years, that's about 40%, but also if you look at 50% survival, that's called the median overall survival. Median overall survival of patients having excluded or desert tumors is about 8 months. Median overall survival of patients having the inflamed tumors is about 13 months. So clearly very important to understand mechanisms that lead to inflamed tumors, and conversely, how do we convert these excluded and desert immunophenotypes into the inflamed tumors? So we have done many studies on this across many tumor types. I'm just going to show you one example of bulk genome-wide RNA sequencing studies we've done with a trial called IMvigor210. That's a phase 2 bladder cancer study, several hundred patients. And this is basically a heat map showing with the red is pathway. So on the x-axis are every row is a gene, and every column is a bit different patient. And you can see on the top, the left, basically 1/3 of the heat map is immune cold, the desert tumors. The middle part is the excluded tumors, and the right third basically is the immune hot or the inflamed tumors. And what you can see here is that if you look at there are the 3 bottom areas here. These are pathways that you can see these first two stars, pathways that are highly upregulated in the immune cold or desert tumors, and much, much lower in terms of activity in the excluded and immune hot. So our question was, what is going on in these pathways, and are there new therapeutic vulnerabilities that might be tractable if we can understand these and be able to convert these immune cold tumors to the immune hot tumors. And you can also see the last star here is a pathway I'll talk about today, the TGF-beta pathway, which is a cytokine involved in the stroma. And you can see here highly upregulated in the excluded phenotypes, but much less pronounced in the immune cold and immune hot. So we wanted to ask ourselves what's going on there in these excluded tumors. And what we published this in Nature a few years ago. We actually looked at our data sets. And this is a great example of how we can use data science to really inform our drug development activities, because we asked the question that if you just look at all the patients, the several hundred patients in this Kaplan-Meier curve, and you actually look at the different quartiles of expression of TGF-beta, you'll see that the blue curve here has the best overall survival. That's the ones who have the lowest expression of TGF-beta. And conversely, if you have the highest expressions of TGF-beta, quartiles 3 and 4, you can see those are the poorest survival. They have the poorest overall survival at 24 months, and they also have the poorest median overall survival if you take the 50% probability. So we wanted to see about if this is really correlative or this is really causative in terms of TGF-beta. I will say also, the question we wanted to ask ourselves was that this is the overall patient population here on the left graph, but if we also looked at, does this make a difference in desert or excluded or inflamed tumors, we looked at those separately, and we looked at the responders, which is otherwise known as people having a complete response, CR, or partial response, PR, versus the non-responders, people who have either stable disease, SD, or progressive disease, PD. So if you look at the box plots here, we looked at basically, we generated a gene expression signature we call the fibroblast TGF-beta response signature, and that's shown in the y-axis. And this is basically a surrogate for TGF-beta pathway activity. So the higher the score on the y-axis, the more the pathway is actually active. And what you can see here is that if you just look at the desert tumors, there's no statistically significant difference between responders, those having a CR or PR, or the non-responders, those having a stable disease or progressive disease. Similarly, with the inflamed tumors, no statistically significant difference. However, if you look at the excluded immunophenotypes, those where the T cells have not infiltrated the tumor but actually are excluded on the perimeter, you see now a statistically significant difference between responders and non-responders in the sense that the non-responders having SD and PD have a higher pathway activity and a higher score. So clearly there is something going on with the TGF-beta pathway driving resistance. So we wanted to see once again if this is causative and actually could we reverse that. So we developed preclinical mouse models which recapitulate the immune excluded phenotype. And what you're seeing here on the graphs is data from our mice experiments. If you have an isotype antibody as a control, you can see that on the x-axis is days, and on the y-axis tumor growth. You can see significant tumor growth. This is the log2 scale, by the way, on the y-axis. So significant tumor growth over time. If you have only an antibody which is an antagonist to PD-L1, you can see that there is some complete response, but by and large, there's still there is certainly not regression in the vast majority of mice. If you only have a molecule antibody against TGF-beta alone, again, looks very similar to isotype. However, if you combine these two molecules, anti-TGF-beta and anti-PD-L1, you now see much more significant complete responses and tumor growth, suggesting that this is, first of all, a causative pathway, and if you can combine using a combination chemotherapy like this, you will actually be able to affect a better outcome. So we are now putting TGF-beta antibodies into human clinical trials, and we hope to see some fruits of that effort in the coming years. But this is a great example of what we call reverse translation, where we can use data science to mine clinical and biomarker data from large phase 2 and phase 3 data sets and inform the discovery of new targets and combinations. Similarly, we've also looked at another cytokine, IL-8, and we've seen, again, this is another example from bladder cancer. This is from our overall survival in IMvigor211, which is a phase 3 bladder cancer trial. This is actually a trial which was randomized. Patients with bladder cancer were randomized to obtain either chemotherapy or atezolizumab, which is our anti-PD-L1 agent, atezolizumab monotherapy. And what you can see here, first of all, there's 4 curves here. Again, this is a Kaplan-Meier curve looking at overall survival. Time is on the x-axis, survival on the y-axis. And you can see here that if you just look at the chemo arms, so the dark blue and the light blue, you can see that patients who have lower IL-8 levels, plasma IL-8, actually have better survival. And similarly, people who have lower IL-8 levels, if you just look at the atezo arms, also have better survival probability. So this is a prognostic biomarker, meaning that it is a biomarker that can be predictive of good outcomes regardless of which therapy is using. In addition, if you just look at the atezolizumab, the 2 arms there, comparing the low IL-8, plasma IL-8, and the high plasma IL-8, you can see that people who actually have low plasma IL-8 fare better after atezolizumab treatment. So this is also a predictive biomarker. So the question we wanted to ask is, are there specific cell types in the tumor microenvironment that are actually secreting this IL-8, and could we understand if there's a predictive aspect here as well? And so we did single-cell RNA-seq. In this particular case, we used 5 responders in this trial and 5 non-responders as an illustration, and we isolated the peripheral blood mononuclear cells, the PBMCs, so we could do single-cell RNA-seq on those on the cells. And you can see in this graph here, we actually can very clearly delineate cells of myeloid lineage from cells of lymphoid lineage. And then when we looked at IL-8 levels, otherwise known as CXCL8, we can clearly see a couple of things. First of all, that the expression is predominantly, almost primarily, in the cells of myeloid lineage. And more importantly, the non-responders actually had much higher levels of expression of CXCL8. So this is suggesting to us that this is a very important biomarker and could potentially predict response in the sense that if you had high levels of IL-8, that are secreted, you're more likely to be a non-responder and less favorable prognosis in terms of treatment from atezolizumab monotherapy. So our hypothesis now, this is really hypothesis generating, is really to now we actually are generating an antibody that can serve as an antagonist and bind IL-8, and we're going to put that in human clinical trials along with atezolizumab monotherapy and see if the combination therapy can once again generate better outcomes for patients. So I'm just going to take a step back. That's a little bit about how we can use data to inform our next studies in terms of understanding mechanisms of resistance. But the other way to actually impact cancer immunotherapy outcomes is to think about the basic mechanism by which cancer immunotherapy is actually acting, and how do we actually, in those patients where it actually is working, how do we improve those responses further? So it sounds like a basic question, but we wanted to ask, how well do we really understand the mechanism by which these anti-PD-1 and anti-PD-L1 drugs are actually working in the body? And I'll say that sounds trivial, but actually our initial hypothesis as a scientific community perhaps 5-10 years ago was a simple reversal of T-cell exhaustion at the site of the tumor. But there is a large amount of data that is now emerging. Ira Melman at Genentech, one of my colleagues, published in Nature Cancer, and there was a group in the Netherlands that published in Cancer Cell, that actually the tumor draining lymph node is a very important site of potentially additional mechanism of action of these anti-PD-1 and anti-PD-L1 drugs, in the sense that dendritic cells, which are present in the tumor draining lymph nodes, also express PD-L1s. And so some of these antibodies may actually disrupt the PD-L1, PD-1 interaction that occurs between dendritic cells and naive T cells in the tumor draining lymph nodes. And by disrupting that interaction, you may actually get better priming of naive T cells, which can then go on to the tumor site. So that's one potential additional mechanism of action that emerging data is pointing to. Additionally, there have been several reports in the last couple of years about a very specific subset of T cells called the TCF1 positive T cells. This is a transcription factor which is associated with stem-like memory cells. And what Nir HaKohen at the Broad Institute did a very nice study where he did single-cell RNA sequencing of melanoma patients. And this was actually tumor biopsies. And what he showed here, and this is just the heat map showing that he actually found two different states of CD8 T cells. He saw one state, and you can see here on the first two rows, TCF7, very high expression, and IL-7 receptor as well. So one state actually had very, very high, TCF7 is the gene that encodes for the protein TCF1. It's a little bit confusing. But so there you found two different states of T cells, CD8 T cells, one that it was highly expressing of these TCF7 transcription factors and were these basically stem-like memory cells. And then you'll see another state here that had no, essentially very, very limited expression and no expression of TCF7. And what they found was that if you actually look at survival, again, this is a Kaplan-Meier survival curve, where you're looking at time on the x-axis, percent survival on the y-axis. This is a small study, but he actually looked at a ratio of these from the single-cell data. What is the ratio of patients who have TCF positive T cells to the ratio of TCF negative T cells, and this is all of the CD8 positive T cells. And you can see here by the two lines here, the patients who actually had a higher ratio, meaning very high levels of TCF7 positivity, and these are the stem-like memory cells, had much, much better outcome than those who did not. And this was further confirmed by research from Arlene Sharp at Harvard Medical School in collaboration with Nick Haining, who showed similar data. They also did single-cell RNA-seq, again, in melanoma patients. They showed here on this graph here, scatter plot, they looked at correlation of progression-free survival, PFS, in these melanoma patients versus the frequency of TCF1 positive, PD-1 positive CD8 T cells. And what they saw here is a fairly good R-squared correlation of 0.49, highly statistically significant, where the higher the amounts of these TCF1 positive cells, the higher the longer the progression-free survival. And they showed that also here in terms of the survivals plot, that patients who actually had the higher ratio of TCF1 positivity actually had high ratios of these, had a very good overall survival. And the authors actually proposed that if we could find methods to actually increase the survival and expansion of this stem-like memory cell subset, that might actually lead to better responses in cancer immunotherapy. So the question becomes, how do we actually do that? So just taking into all this data into account, one model that is emerging, and I will just say this is one model, there are probably multiple mechanisms by which these anti-PD-1, anti-PD-L1 drugs are working, is that instead of thinking about this simply as reversing exhaustion, you can think of it as potentially expanding a stem-like memory cell population in the tumor cells, in the sense that these TCF1 positive stem-like memory cells interacting with dendritic cells. The interaction is the PD-L1, PD-1 axis is interrupted by these checkpoint blockade molecules. That allows these TCF1 positive memory cells to actually expand, go to the tumor, to actually become effector T cells, and then after prolonged antigen exposure, they become exhausted and hyper-exhausted T cells. So that is one potential mechanism that is emerging in the field. And so the question again becomes, how do we further investigate this, and what are mechanisms, if these TCF1 positive cells really are the effector molecules and the progenitor molecules, how do we actually expand their population? And I will say that there are two highly expressed checkpoint molecules on these TCF1 cells. One is PD-1, and another one is a new molecule target that was found at Genentech a few years ago called TIGIT, which stands for T-cell immunoreceptor with immunoglobulin and ITIM domains. And basically what TIGIT is a receptor molecule on T cells, and it competes with another molecule, CD226, for binding of its ligand, PVR, also known as CD155. So if TIGIT binds its ligand, PVR, it basically prevents the ligand from binding CD226, and that prevents CD226 from activating T cells and affecting an immunotherapy response. So this axis of PVR TIGIT is very, very relevant for immunotherapy and potentially tractable as a drug molecule. And we've shown that by a randomized phase 2 study where we randomized patients to receive either Tecentriq, which is atezolizumab, our anti-PD-L1 monoclonal antibody, or the combination of Tecentriq, atezolizumab, plus tiragolumab, which is our anti-TIGIT molecule. And the hypothesis is that if this TIGIT PVR axis is important, then a combination molecule, a combination therapy of Tecentriq plus tiragolumab, in other words, anti-PD-L1 and anti-TIGIT, would have greater benefit than just the Tecentriq alone. And we looked at this in frontline non-small cell lung cancer. And so the bar graphs here are showing the response. And so the graph on the left-hand side is the intent to treat population. So this is all 135 patients. We see a modest improvement in the response rate. If you look at the combination of tiragolumab and Tecentriq versus placebo plus Tecentriq. But interestingly, if you look at a biomarker called PD-L1, which is basically a surrogate for an inflamed microenvironment, you see a very dramatic increase that tiragolumab plus Tecentriq, response rate of 66%, which has not been seen before in frontline lung cancer. And the placebo plus Tecentriq in the same population is 24%. And you can see that this is obviously the high PD-L1, in other words, those patients who have an inflamed tumor microenvironment, is driving all of the response, because if you look at the third graph here, PD-L1 moderate 1 to 49%, in other words, moderate staining of the tumor cells, they have basically no improved benefit of the combination. So goes to show us that the combination here of tiragolumab and Tecentriq can have very meaningful clinical benefit, and that this TIGIT PVR axis is very important. And this is further demonstration of that. If you look at the Kaplan-Meier curve, this is just progression-free survival. You can see that in the blue line here, median overall survival only about 4 months if you just have the anti-PD-1 by itself. But the red line here, if you look at the combination of anti-TIGIT and anti-PD-L1, has a median PFS, progression-free survival, not reached, but a very, very meaningful hazard ratio of 0.3. Hazard ratio is basically what is the reduction in the risk. So you have a 70% reduction in the risk of a progression-free event, of a progression. So very, very impactful axis here now, PVR and TIGIT, and we are taking this this data into a large phase 3 global study. We already have started this in frontline non-small cell lung cancer, and based on PD-L1 expression and TIGIT expression, we're also looking at many, many other different indications such as cervical cancer, small cell lung cancer, et cetera. So this is one example of how we can start to mine data and think about mechanism of action and how that might actually inform a new combinations. So further evidence that this TIGIT PVR axis might be important in patients who are actually undergoing treatment with cancer immunotherapy and specifically PD-L1 or PD-1 blockade was actually presented by a group of investigators and published in JCI Insight last year. They actually looked at it from the biomarker perspective. And as many of you probably know, PD-L1 is basically, as I said before, a surrogate of an inflamed tumor microenvironment. It's not a very predictive biomarker in the sense that you can see here in these bar graphs, if you just look at the bar graph on the upper left here, this is looking at responders, so people who are responding to anti-PD-1 therapy, and the non-responders as in gold here. And you can see that if you have high PD-L1 levels, you do have a good response, but you also have non-responders. And conversely, just having a low PD-L1 level, you still have people. You have 10 patients here in this particular example, so about 25% response rate, even in the low PD-L1. So it's not a very good biomarker. They also showed that PVR, which is again the ligand for TIGIT, also is not a great biomarker in the sense that if you had high levels of PVR, you still had some response, and the low levels you had moderate response. But the investigators interestingly looked at, if you ask the question, what if you had looked at both biomarkers simultaneously? And what you'll just see, if you just focus on the last bar here, if you actually had low PD-L1 levels, so indicative of basically of not a very inflamed microenvironment, and high PVR levels, you didn't get a single response. The response rate was 0%. So again, some data suggesting that the combination of low inflammation, inflamed T cells, and this PVR TIGIT axis might actually predict response and might actually tell us that, again, that axis is very important. And you can see that, again, by the Kaplan-Meier curves. They looked at progression-free survival. If you look at the top curve here, the difference in PD-L1 and PD-L high and low is basically what we've seen before, that people who do have high levels of PD-L1 in general have better outcomes excuse me, from cancer immunotherapy. But you can see the median overall survival, not a huge difference, statistically significant, but not a clinically meaningful, not a very clinically meaningful difference in terms of about 2 months for the PD-L1 low and 5 months. Similar data for the PVR, but if you look at the bottom graph here, you can actually see that people who have high PD-L1 and low PVR, 13-month median progression-free survival versus the opposite case, PD-L1 low and high PVR, only 1.4 months in terms of median PFS. So further data suggesting that this axis is very important. So I'm going to switch gears a little bit now to talk about how we can further improve responses in cancer immunotherapy. Again, this is another example of how we can use data science. And so one thing we've noticed in many of our studies is the presence of something called a tertiary lymphoid structure. And that is, as many of you probably know, these are organized aggregates of immune cells outside of lymphoid tissue. So these are aggregates, as this picture is showing here, that are present in the tumor site. And what we've seen across many of our studies, this is one example on the right-hand side of a Kaplan-Meier curve in non-small cell lung cancer. We saw that in our randomized study, for patients who actually had a tertiary lymphoid structure present, had significantly better overall survival in terms of atezolizumab treatment, but the same was not true for chemotherapy. In other words, this was not a prognostic biomarker, but very predictive for better outcome with atezolizumab treatment. And we've seen the same types of data across bladder cancer and breast cancer and many other cancers as well. So there's something very important here about the presence of a tertiary lymphoid structure. And I will note that Jennifer Wargo at MD Anderson also published in Nature last year, similar data in melanoma, where she also showed that the presence of a tertiary lymphoid structure induces better response for these anti-PD-1 treatments, in her case, melanoma. So we are looking at this. I think this is an opportunity to look at digital pathology. So can we actually use machine learning and AI to actually identify and extract features in histology images that are associated with the tertiary lymphoid structures and generate an algorithm so that we can robustly and on a large scale actually identify these tertiary lymphoid structures, and maybe even mechanistically try to understand how these tertiary lymphoid structures are actually imparting a better clinical efficacy in our cancer immunotherapy programs. And can we actually find new targets? And we are actually looking into this now. We found a potential 2 or 3 potential targets that we're now validating that if we can generate agonists for those targets, they can induce these tertiary lymphoid structures. And we hope to see now a combination therapy of an agonist with atezolizumab will actually impart a greater clinical efficacy after cancer immunotherapy. I will also say that this is also an additional opportunity for spatial transcriptomics, because if we can start to understand, what are the specific transcriptional programs that are in these immune cells, whether it's the B cells or other cells that are in these tertiary lymphoid structures, if we can better understand the transcriptional programs that are present in these immune cell subtypes and specific subtypes, we can also find new targets there as well. So I'm going to talk a little bit now just about the sort of integrated view of mutations, somatic mutations in tumors, and how that actually affects cancer immunotherapy response. So we also did a study we published in New England Journal of Medicine, I believe last year. We called it the Empower 150. This was a large global phase 3 study for non-small cell lung cancer, where patients were treated with either chemotherapy or chemotherapy plus atezolizumab, our anti-PD-L1 agent, plus bevacizumab, which is an anti-VEGF, angiogenesis molecule. And we found it was a positive trial, so we found very favorable outcomes if people were actually treated with the combination of chemotherapy, atezolizumab, and bevacizumab. However, in the general population. However, we did find this one subset of patients with non-small cell lung cancer who had either STK11, which is a tumor suppressor, and or mutations in KEAP1. And that's shown here on the bar graphs here, on the Kaplan-Meier curves here, progression-free survival and overall survival. You can see that if you have no mutations in either of these genes, there's wild type for both STK11 and KEAP1, that's the black line here. They actually have higher median overall survival, about 20 months, and higher landmark survival at 2.5 years. But in contrast, if you have a double mutation, in other words, patients who have mutations in both STK11 and KEAP1, that's the blue curve here, they have the poorest overall survival and progression-free survival. So we wanted to ask ourselves, what is going on in the tumor microenvironment? What are these mutations actually conferring to the tumor and to the microenvironment? And is there something we can understand mechanistically that then can lead to actually having an additional therapeutic modality that might actually improve their outcome? So we did RNA sequencing on patients who had either double wild type, in other words, neither of these mutations, or had both of these mutations, just as the two extremes. And this is what is called a volcano plot for obvious reasons. I'll orient you. On the x-axis is basically the log2 of the expression change. In other words, what is the relative expression difference in the double mutants versus the wild type in the wild type for a specific gene, and each transcript is denoted by a dot here, a red dot. And on the y-axis is the negative log 10 of the p-value. So the higher the value, value on the y-axis, the more highly statistically significant this is. So just to orient you, a p-value of 0.01 would be basically a 2 here on this y-axis. So what we're finding here is that, very interestingly, if you just ask yourself the question, in these patients with double mutants who had very poor outcome after cancer immunotherapy, we're seeing a lot of transcripts which are actually associated with a program called ferroptosis. And I'll just point you to a couple of ones here, the SLC7A11 and SLC3A2. And there are also all these AKR gene transcripts, and they are all involved in either ferroptosis or reactive oxygen species here. What is actually going on here? Well, at the same time we actually found this data, Charlie Rudin at MSK also published very similar data. He also had a similar data set in non-small cell lung cancer, and he actually did a lot of validation studies and showed a very nice model that for patients who actually have only a KEAP1 loss or only STK11 loss, you get protection from ferroptosis. And ferroptosis, I should point out, is an iron-dependent programmed cell death module that is distinct from apoptosis and distinct from pyroptosis and necroptosis. So it is a form of programmed cell death that is highly dependent on iron and involved in the lipid peroxidation. Now, what he also showed was that for patients who had the double mutations, STK11 and KEAP1 mutations there, so these are loss of function mutations, they actually had very significant protection against ferroptosis, and you could actually treat these patients with an inhibitor to SCD1, and they would actually undergo ferroptosis. So what you may be asking yourself, what is the relationship of ferroptosis to the cancer immunotherapy, and specifically to these mutations? So there is a very nice study that was published in Nature by Arul Chinayan and Weiping Zhou at the University of Michigan a couple of years ago, that actually showed that ferroptosis is an underappreciated and perhaps to date unappreciated mechanism by which T cells actually mediate anti-cancer immunity, in the sense that T cells are known to release interferon gamma, and what interferon gamma does to the tumor cell is it downregulates SLC3A2 and SLC7A11, which are two components of the glutamate cysteine antiporter system. And what that does is it actually downregulates the uptake of cysteine by tumor cells, and that leads to lipid peroxidation and ferroptosis. And they showed this very nicely with patients who were melanoma patients treated with anti-PD-1 drugs. And I'll just orient you to these are violin plots here. What they showed is that on the first plot here, this is simply looking at SLC3A2, which is one of the components of this glutamate cysteine transporter. And in patients who had clinical benefit, that's the one on the left here, you can see that they actually had a reduction, statistically significant reduction in the SLC3A2 transcript, and they also had an increase in interferon gamma, as you would expect. And conversely, people with no clinical benefit, and this is actually looking at after cycle 2 versus a baseline. So this is the change in this transcript after drug treatment. So Very nice data showing that you can that interferon gamma, in addition to acting as a in a more traditional way as a cytotoxic agent, it also downregulates this glutamate cysteine transporter system and results in ferroptosis, which ultimately results in cell destruction. So basically, what we're finding is that these mutations that the tumor cell induces, in this particular case, a double mutation of STK11 and KEAP1, result in a protection against ferroptosis, and that protects against interferon gamma. But you can, as Charlie Rudin showed, you can now look at new Achilles heels or therapeutic vulnerabilities that are created by this particular pathway. So this has opened up a new area for us to look at in terms of new therapeutic modalities. And again, this is based on looking at mining clinical and biomarker data from our large trials and understanding what's mechanistically driving resistance. So I'm going to switch gears in the last few minutes of the talk and talk about circulating tumor DNA as a diagnostic biomarker and a predictive biomarker. So I'm sure a lot of you in the audience are familiar with circulating tumor DNA. It is basically, as the name implies, DNA that is presumably released by apoptotic cells, tumor cells. It has a very short half-life of less than a couple of hours in circulation, but it can be now, given the advent of high sensitivity next generation sequencing and computational approaches, we can now identify and track and quantify levels of circulating tumor DNA. And as this graph is showing, typically it is a very nice surrogate for tumor burden, and much like PSA in the metastatic setting for castration-resistant prostate cancer. So what this graph is showing is that, after surgery, you will sometimes release amounts of circulating tumor DNA into the circulation. Once you have a successful adjuvant treatment, the levels of circulating tumor DNA go into very low amounts, but once the tumor starts to generate resistance, you will have a relapse, and that relapse is preceded by this increase in the circulating tumor DNA amounts. And if you can actually treat successfully in first-line metastatic setting, advanced disease, you can actually see a reduction in circulating tumor DNA. So we've done a lot of work in the metastatic setting using circulating tumor DNA as a biomarker to predict the onset of resistance. We've also used it as a non-invasive biomarker to look for resistance mechanisms. But I'm going to talk in this last few slides about the use of circulating tumor DNA in the adjuvant setting, the earlier settings of disease. And people have shown that circulating tumor DNA in the adjuvant setting can serve as a very useful prognostic biomarker because, not surprisingly, if it serves as a surrogate for tumor burden, then it goes to it suggests that if you actually have low levels after surgery, you're probably cured by surgery. But if you have detectable levels of circulating tumor DNA, you probably have micrometastatic disease and a much higher risk of relapse. And in fact, many investigators have shown that, in fact, if you have detectable circulating tumor DNA, you actually have higher risk of relapse, and you do relapse much earlier, and that's across many different tumor types: breast cancer, colorectal cancer, and lung cancer. We wanted to use this in an adjuvant setting, knowing that high levels of circulating tumor DNA would predict a relapse. We wanted to ask the question, in the adjuvant setting, could circulating tumor DNA serve not only as a prognostic biomarker, but as a predictive biomarker to predict patients who will respond to a cancer immunotherapy, in this particular case, atezolizumab. So we had a phase 3 global trial we called IMvigor010. This is the adjuvant study in muscle-invasive urothelial cancer, so bladder cancer. The key eligibility criteria is shown on the left. This is for people who have high-risk muscle-invasive bladder or upper tract cancer. They actually had radical surgery, and they had tissue sample available. And these people were randomized 1-to-1 to either have observation only or atezolizumab. And unfortunately, and the endpoints were disease-free survival as a primary endpoint, and secondary endpoint was overall survival. Unfortunately, this trial did not meet its primary endpoint of disease-free survival in the intent-to-treat population. In addition, the more traditional biomarkers, PD-L1 and tumor mutation burden, did not identify patients who would benefit from atezolizumab in this particular indication. We did, however, do a pre-specified analysis of circulating tumor DNA in these settings. In other words, we did a prospectively defined retrospective analysis, and we asked the question, if you looked at circulating tumor DNA, was it prognostic and predictive for response for atezolizumab? And that data is shown here. These are Kaplan-Meier curves, and I'll just focus on the right curve. That's overall survival, but the same trend is apparently disease-free survival. And what you'll see here, if you just look first of all at the observation arms, you'll see that if you have circulating tumor DNA, that actually, as people have seen before, portends for a worse prognosis, in the sense that this curve here in the dark red is actually much worse than if you have no ctDNA present. Now, so it's certainly prognostic. And if you look at now the blue curves here, the presence of circulating tumor DNA and atezolizumab treatment, you can actually see a hazard ratio of 0.59, showing that and an improvement in median overall survival. And this is the first data showing that the presence of ctDNA is not only a prognostic biomarker for outcomes, but is also can be used as a predictive biomarker in the adjuvant urothelial cancer setting to predict those patients who will actually have a better outcome after atezolizumab treatment. And the same is not true for if you have no detectable ctDNA. You can see there these two curves at the top are basically overlapping, hazard ratio not statistically significant of 1.31. So no benefit if you have undetectable ctDNA, but if you have detectable ctDNA, it serves as a predictive biomarker, and you will have better outcomes with atezolizumab treatment. And so one of the questions we asked ourselves is, because we had tumor tissue from these patients, we could ask ourselves, what are some of the transcriptional programs that are actually being upregulated in people who actually have ctDNA and are relapsing earlier? So we looked at, just in the observation arm, people who are, in this particular case, do not have any detectable ctDNA, because you'll recall that even those people who do not have detectable ctDNA, that doesn't mean they don't have ctDNA that is circulating. It just means we could not detect them in this particular assay. So we asked ourselves, what are some of the transcriptional programs and pathways that actually separate relapsers from non-relapsers? And that's shown in this gene set enrichment analysis here. Those pathways that are upregulated in the relapsers are shown here in the bottom in red. You'll see TGF-beta and epithelial mesenchymal transition. So clearly, those patients who have upregulation of these pathways, among many others, they are at higher risk of actually having relapse, even though they have no detectable ctDNA. So we're diving into this data a bit further, and we're finding potential targets that could be potentially used, again, in addition with atezolizumab, to actually delay the onset of relapse in these patients who have no detectable ctDNA. And similarly, we could also ask the question, in those patients who are ctDNA positive and are treated with atezolizumab, we called from the Kaplan-Meier curves, it was a predictive biomarker, and patients did have a better outcome, hazard ratio of 0.59, but even there, we have patients who relapse. And once again, we can do gene set enrichment analysis. We're seeing TGF-beta. We're also seeing angiogenesis signatures arising there. So once again, showing a power of data science to actually point to further, in terms of directionality, further pathways to look at in detail to find new targets and combinations that might actually help improve cancer immunotherapy. And lastly, my last slide, we also asked the question, well, ctDNA is a great biomarker at baseline to predict response or predict prognosis. Can we also use circulating tumor DNA as a dynamic marker, much like, again, people use PSA changes and increases and decreases? And does that change in ctDNA actually predict response? In other words, if you take a baseline measurement of ctDNA and then you look at cycle 2, day 1, or cycle 3, day 1, so after 3 or 6 weeks of treatment, do you see decreases in ctDNA, and does that decrease actually correspond to survival? And what we showed here in this bar graph on the left-hand side is that people who are treated with atezolizumab, you can see, have a highly statistically significant difference in ctDNA clearance. In other words, they have a higher ctDNA clearance meaning their baseline was positive detectable ctDNA, now going to negative undetectable ctDNA. So they're clearing the ctDNA, and that is associated with atezolizumab response. And you can see that very clearly in the Kaplan-Meier curves. Again, I'll just focus on overall survival on the right-hand side. Those people who have a ctDNA clearance, in other words, were positive at baseline, now negative after just 2 cycles of treatment, now have a very statistically significant, meaningful hazard ratio in terms of both overall survival and progression-free survival. So ctDNA can be used not only as a baseline marker to predict prognosis and therapy response, but also as a dynamic marker on treatment as an early efficacy readout to predict treatment. And the reason why this is important is early on, we can start using this now in our clinical development studies to do faster clinical development using ctDNA as an early readout to say, yes, this combination is going to have a meaningful benefit. Let's take it very quickly into a global, large phase 3 registration study. We can also imagine a scenario down the road, maybe 5 or 10 years, where this is a surrogate for disease-free survival or even overall survival, surrogate meaning a regulatory endpoint that the FDA will approve. And instead of waiting 10 years to do an adjuvant study, we can do a quick readout in a few months and actually get important drugs on the market faster. So these are just some examples of how we're using our computational approaches. And with that, I'll just summarize. Hopefully, I've given you a few vignettes of how we're using computational and data science to understand mechanism of action and also inform our understanding of resistance mechanisms and therefore targets and combinations. Just one example of how tumors with specific mutation profiles can create new therapeutic opportunities, and would love to talk to you again about some of the other mutations that we're seeing. BRAF, V600E is a great example, and how that's actually inducing protection from pyroptosis. There's some very interesting data emerging there. And lastly, we really think that we're in the very early days with computational, and these single-cell approaches are really providing a very, very nice granular view of tumors, but immune cell subtypes, and which it's going to be very important to say not just infiltrated by T cells, but which types of T cells, and are they tumor reactive? And that's really going to help us direct some of our therapies to patients in need. So with that, I will thank you, and let's take any questions.
Ron, thank you so much for the lovely presentation. You've given us a lot to think about, certainly. I'd invite people to ask a couple of questions. We have some time for questions. You can either sort of raise your hand, or you can type your question directly in the chat, and I'm happy to relate it.
I'm going to stop sharing if that's okay.
Yeah, please go ahead. I can start it off with a question. So Ron, I guess one of my especially about your last vignette there, would you suggest that this is be something implemented? I mean, you were talking about it focused on trials, but would you suggest that monitoring of ctDNA levels could be something as sort of like an early warning for if chemotherapies are working, or if you need to sort of change course early? Is that, I'm not asking you for a clinical recommendation. I'm just asking you sort of your thought process from your looks at this.
Yeah, I will say that I am cautiously optimistic. I will say that, and I didn't mean to oversimplify things, but just in the interest of time. But I will say that, as with any biomarker, you want to be absolutely sure that what is the prevalence of the biomarker, how dynamic of a change is that, and does that really correspond to survival? So, for example, in some of our non-small cell lung cancer study, we're seeing a much lower prevalence of ctDNA. And I think how predictive it is. We are generating data. We just had our first, this is public release now. We have a adjuvant study in non-small cell lung cancer, which was positive. We're looking at ctDNA and how it predicts, how changes in ctDNA predict overall survival and disease-free survival there. So I am cautiously optimistic. I think the short answer to your question is yes. However, I would just say that we need a lot of validation just to prove that not only in bladder cancer in the adjuvant setting, but also in lung cancer in the adjuvant setting with a cancer immunotherapy drug or a combination or targeted therapy, that this will indeed be useful. So I hope the answer is yes, but I would like to just caveat that by saying we need some more data.
I guess it suggests that there should be large-scale trials and studies where we're looking for this, we're monitoring this in different forms of cancer to see how informative it can be as paired with the Kaplan-Meier. I mean, the Kaplan-Meiers that you showed were just pretty exciting in terms of like the differences that was observed.
Yes.
I certainly don't mean to dominate. Does anybody have questions? Please type in the chat. If you have a question or so, or again, you could raise your hand. I'm sort of looking at the participants list. I'll give it a minute. I found teaching class, Ron, that sometimes you got to just be quiet and let that awkward silence stretch to get the questions.
Winston, can I make a comment before?
Please go ahead. Go.
Ron, thank you. That was really great. I was really glad to see you had spatial transcriptomics, in your last vignettes. Really important. We think it's really important. Certainly, biomedical engineering has recruited numerous people in it, and many of us are working in it now. So really great to see, how that's evolving at Genentech and in your group. So thank you so much for that talk today.
Yeah, thank you.
Thanks, Michael. So we do have a question from Yunan, who's asking what current technical advances can be used to sort of improve problems with sequencing circulating tumor DNA, and accurate quantification. As I'm sure you know, Ron, it's a bit of a challenge, right? Because first of all, there's not that much of it, and second of all, it's often pretty degraded. And so I'm curious what your perspective is at Genentech. I'm paraphrasing her question and then adding to it a little bit. What sort of your perspective is at Genentech on this problem?
Yeah, I think it's a great question. So I think we need we still need higher sensitivity methods, first of all, and I would also argue that we need methods that are customized on a sort of almost personalized level, in the sense that no two tumors are alike, right? One patient may have a certain set of somatic mutations because they've undergone various selection pressures and treatments. Another patient, even the same cancer, may have a different set of mutations because of the tumor microenvironment, selection pressures, treatment history, et cetera. So I think a couple things. One is we have to really think about how do we actually develop more customized panels or assays that are customized to really detect that patient's tumor profile, and then just higher sensitivity assays. I mean, the question really is going to become, is it biology limiting or technology limiting that we can only detect, even in the bladder cancer example I showed you, only 37% actually had detectable ctDNA. So I don't know if that's real or that's just an artifact of our sensitivity. So I would just and then just lastly, just as it was alluded to in the question, just robust methods to make sure that we can actually get the plasma and get as much circulating tumor DNA out of the plasma as possible on a reproducible and robust basis. So I think certainly these are all good proof of principle type studies I presented, but I think the field is moving very quickly towards that.
Yeah, I would agree. So there's another question here from Amelie S. And asking, what are some of the breakthroughs in the digital pathology in the pharmaceutical industry, the cancer mutation patterns, given tools like machine learning, data science, and computational biology?
Yeah, that's a very open.
A bit of a broad question. Yeah.
No, I think it's a great question. I think I will say that there are. I think we have with these new tools, if you can combine histology and digital pathology and machine learning on histology images, so that you can actually derive spatial information and correlate that with, for example, TCR sequencing or immune cell subtypes and mutation profiling. It gives us a much more complete picture. So I would just say that an integrated view that combines all of these different orthogonal and rich data sets is going to be much, much more informative in the future than just tumor mutation profiling. So I think, before, 10 years ago, we had, people were looking at 60, or now the Foundation One Medicine panel has 324 genes where you have whole exome coverage. Now we're looking at whole exome sequencing. I think the more information you can actually ascertain and mine with good algorithms and machine learning tools, the better your chance that you'll actually find something more useful. So I don't know if that quite answered the question, but I would just say that we have to put all these tools together.
Yeah, I think that's it's a tough question to answer. I think that was an excellent job at that. Maybe we can have time for one more question.
I have a quick question.
Go ahead, Jeff.
Oh, but I will defer to Peng Fei.
Okay, Peng. So the question from Peng is: could you say a little bit more about how you think spatial transcriptomics is going to help, especially as resolution's increasing? 10x is now saying they're going to go down even smaller. Of course, Aviv has shown data where she's going even smaller and smaller in spatial transcriptomics. Again, I'm expanding a bit on the question, but how do you think it's going to help?
I think it's going to help because one thing we don't understand is proximity of myeloid cells, so macrophages, tumor-associated macrophages, for example, and there are, as a simplification, there are sort of the M1, anti-tumorigenic and M2-like pro-tumorigenic macrophages. Understanding not only which of those are maybe in close proximity to a tumor cell or other immune cells, and how that's related to response to our cancer immunotherapy drugs, but also even subtypes of M1 or M2 and transcriptional programs there that might actually also be very important for in the immune synapse, and how that's important for destruction of tumor cells. And so I think as much information we can glean to say this specific immune subtype with this is enacting this transcriptional program, and at this point in time, that is making a difference because it is having this effect on cancer immunotherapy. So I think when we can get down to that detailed sort of spatial and temporal level, that's when we really start to understand a lot of our drugs in that data.
And so there are some more questions, but I'm afraid I'm going to have to pass it over to Jeff to close because we're already a little bit past time. Jeff, do you want to take it away and give us some concluding remarks?
Thanks very much, Winston. Sure. And I invite everyone to stay on if Dr. Mazumder is willing to stay on for overtime. But Winston, that's a good call. And I might stay on because I have a ctDNA clearance question too.
I can stay on 10 more minutes if that's okay.
Awesome. Thank you, sir. Well, but first, before we adjourn the seminar, I wanted to thank you very much on behalf of the Department of Biomedical Engineering. We're so glad that you came back and that you shared your perspective with us. Thanks very much. And I'm sorry that the resounding applause isn't evident over Zoom, but it was a great presentation. Thanks also for your time this afternoon in talking with faculty, the PhD students. I hope that was good, and I hope some of the PhD students are here. I really look forward.
You should ask them what they think, but yeah, I enjoyed it.
Awesome. Great, thanks very much. And I had we have a small token of appreciation for you, Ron. And just as science is the engine, right, of discovery and advances in medicine, here we've got the official HEMInar token of appreciation, which is a fusion of an engine with the Caduceus. And this is on its way to you, Ron, by the mail. And again, I hope it's a reminder of your days at Johns Hopkins.
Yeah.
And hopefully more of those in the future.
Great, fantastic. Thank you guys for this opportunity, and really enjoyed the day.